Hi, my name is Majdi Masarwi. I'm a postdoctoral research fellow in the Reagan Lab in the Main Medical Research Institute, and my research here focuses on studying multiple myeloma and the role fatty acid metabolism plays in cell growth, survival, and drug resistance in the bone marrow niche. Multiple myeloma is the second most prevalent form of blood cancer, with around 30,000 new cases expected to occur in 2018 in the United States alone. This fatal disease originates from plasma cells, immune cells in the bone marrow that undergo a series of oncogenic mutations. Eventually, the cancer will metastasize in the bloodstream, becoming plasma cell leukemia, causing a thickening of the blood and subsequent kidney failure and anemia. At this stage, the cells are no longer reliant on the bone marrow niche for survival and proliferation. Multiple myeloma also interferes with the normal process of bone cycling. Bones are constantly being remodeled, with cells called osteoblasts laying down new bone, as others, called osteoclasts, resorbing old bone. Multiple myeloma disrupts this balance, leading to an increase in the activation and number of osteoclasts and inactivation and decrease in number of osteoblasts. The bones weaken as lesions start to form. Initially, multiple myeloma is responsive to chemotherapeutic treatments. However, after a period of time, the cells become drug resistant. As of now, there are no effective treatments after this point. Multiple myeloma is found in the bone marrow, presenting the cancer cells with a unique microenvironment. The bone marrow is rich in immune cells, bone cells, mesenchymal stromal cells, growth factors, and cytokines. All of these play a role in the growth, progression, and survival of myeloma. But we are specifically examining are the fat depots composed of bone marrow adipose tissue, also referred as BMAT. BMAT accounts around 50 to 70% of the bone marrow volume. Over the past several years, BMAT has been shown to play an active role in bone metabolism, bone cancer metastasis, and drug resistance. We are examining the role of lipid metabolism in myeloma survival, proliferation, and drug resistance. In vitro co-culture of bone marrow adipocytes and multiple myeloma have shown to support myeloma growth and chemoresistance. We have shown that the adipocytes shrink in response to multiple myeloma cells, perhaps due to lipolysis or another form of delipidation. We're currently examining whether multiple myeloma cells are using the lipids from the surrounding adipose tissue for energy production through fatty acid oxidation or for de novo fatty acid synthesis. Other studies that inhibit fatty acid oxidation in cancers, such as leukemia and bone metastatic breast and prostate cancers, have seen a decrease in cell proliferation and survival. Our goal is to determine the role of lipid metabolism in multiple myeloma cells and how bone marrow adipocytes promote tumor progression and drug resistance in vivo and in vitro models. Understanding the interaction between bone marrow adipocytes and myeloma cells may provide a new area to develop new drugs.